Our genome stores a lot of information. But how is all this information turned into functional units? Our genome is basically a manual that contains different sets of instructions. These instructions appear in the form of genes. When a certain functionality is needed in a cell, genes are activated and they are turned into polypeptides, which will fold into proteins. These proteins are the end products in the cell that carry out many important activities. The production of proteins happens in 3 steps, which together is also known as the central dogma of molecular biology, which was postulated for the first time by Nobel Prize winner Francis Crick. First, a gene is turned into precursor RNA molecules via a mechanism called transcription. Second, the precursor RNAs are processed by the splicing machinery into mature messenger RNA, better known as mRNA. Third, the mRNA is read and turned into a polypeptide chain, which is known as translation. So let's have a look at each of these steps. Gene transcription starts when the gene is turned on in the nucleus. This happens when different proteins, known as transcription factors, bind around the promoter region surrounding the gene, which enable the RNA polymerase II to transcribe the following DNA sequence into RNA. RNA, like DNA, is assembled as a chain of nucleotides. However, it is single-stranded, and each thymine base is substituted by a uracil. This RNA sequence is also known as the pre-mRNA and is processed in the next step. The pre-mRNA still contains the introns of a gene, which, in contrast to exons, do not contain information about the polypeptide. Therefore, they are removed from the mRNA sequence through a mechanism called splicing. The splicing machinery recognizes specific sequences within the intron and surrounding the intron-exon boundaries, and subsequently removes the intronic parts of the gene. The processing of pre-mRNA is not always the same, but can differ per tissue. For instance, specific introns are retained in one tissue, while certain exons are removed in another tissue. One gene can thus produce different mRNA sequences that each produce different proteins. This is called alternative splicing, which adds to the complexity of the biological system. There are more processing steps next to splicing, such as capping or polyadenylation, but those will be covered another time. Finally, the processed mRNA strand is transported out of the nucleus to be translated into a polypeptide chain. Translation occurs either in the cytoplasm or at the endoplasmic reticulum, where different components of the ribosome attach to the beginning of the mRNA strand to initiate the translation process. The ribosome is like a factory that uses mRNA to form the polypeptide chain by linking amino acids one at a time. This occurs with help of specific RNA molecules called transfer RNAs or tRNAs that carry one of the 20 naturally occurring amino acids on one end of the molecule. On the other end, 3 nucleotides are exposed that are complementary to the mRNA, which enable their binding. As a result, any 3 nucleotide sequence, or codon, on the mRNA encodes for a specific amino acid. The codon table provides an overview of the amino acids that are encoded by the codon. The translation starts when an AUG codon is encountered and tRNAs bind to the mRNA and a methionine is built in as the first amino acid. The chain is then elongated by reading the next codon as the ribosome travels over the mRNA strand. For example, if the next codon is AGC, a serine amino acid is added to the chain. If the next codon is GGC, a glycine amino acid is added. The division of the mRNA coding sequence into codons is also known as the reading frame. The elongation is terminated when a stop codon is encountered. The complete polypeptide chain is released from the ribosomal complex, which shortly will fold into a three-dimensional functional protein that will function in the cell. This process occurs constantly in every cell, as all genes that are activated follow the central dogma, and the stored information in the form of DNA becomes functional in the form of proteins. This enables the existence of complex organisms, such as us humans.